Hello, good afternoon, everyone. So let's start by reviewing some of the key things that we learned last time before we continue on. So we learned about the cell theory. So cell theory has four parts to it. Number one, organisms are made of one or more cells. So things could be unicellular organism, which is one cell, like amoeba, like paramecium, or they could be multicellular, like a human being. Second part is that cells contains all the properties of life and it's individually alive, even though they are sometimes part of a multicellular organism. Number three, uh, new cells came from the division of pre-existing cells, and that when they do divide, they will pass on their hereditary material, right? Their genetics information. And so we talk about two kinds of cells, right? They could be eukaryotic cell, or they could be prokaryotic cells. So common features of all cells include cell membrane, um, genetic material, and cytoplasm. These are common features, right? And then we can categorize the cells into eukaryotes and prokaryotes. Here are some of the main differences. For eukaryotic cells, they have a nucleus. And we talked about the nucleus last time. That's a distinct circular structure where you will be able to put your DNA in. So for the prokaryote, no nucleus. Eukaryotic cells tends to be bigger and more complex, whereas prokaryotic cells are smaller and simpler. Also, eukaryotic cells have many organelles. And so we already learned a little bit of organelles last time, right? We learned about the ribosomes, for example. Right? We learned about, the nucleus, the nucleolus. Whereas for prokaryotic cells, there are fewer organelles. And examples of eukaryotic cells include animal, plant cells. And examples here, like bacteria. So that's some of the main concepts that we learned last time. All right, and then we went on and drew a bit of a cell. All right, let me just move this stuff out of the way so we can continue our picture. Okay. So hopefully you still have that picture. Please take it out, and we're gonna add more details to it, soon. Okay, so that was the picture. Let's quickly go through some of the things that we drew. We have the nucleus here, and of course this is a eukaryotic cell, right? So for the eukaryotic cell, That's why it has the nucleus. On the other hand, we have the prokaryotes, which don't. So surrounding the nucleus is going to be this envelope, right? The purple envelope right here. That will help control what goes in and out of the nucleus. But there are some gaps in the envelope. We call them nucleopores. That's actually how things can squeeze through and enter or leave the nucleus. That nuclear envelope extends outward into the cytoplasm. Okay, so all these things here that you see, those are the extension of the nuclear envelope, and some of them will have the ribosomes on them, and then you will have some that does not have the ribosomes on them. Okay, so I haven't really told you about what this is called yet, but I think it's a good time to write it down now. Okay, so this stuff, right, this network with the ribosomes on them, we call this the rough endoplasmic reticulum. Right, the rough endoplasmic reticulum. So endoplasmic means is within the cell, right? Reticulum just means a network. So this is a network of membrane that has a rough appearance to it because of all the ribosomes that are stuck to it. Okay, so RER, that's the short form, rough endoplasmic reticulum. Okay, so what does it do? It actually works with ribosome to synthesize, to make, right? And package proteins. That's what the rough ER is going to do.
Professor Vincent, is it made of protein itself?
The membrane?
Yes.
No, this is not made of. I think you're thinking about the ribosomes.
No, I'm thinking about this reticulum.
Oh, reticulum is not. Reticulum is mostly just membrane, so like lipids.
Lipids.
Yeah, the phospholipids. Yeah. Okay, so I'm gonna show you how this works. Okay, it's a little bit weird. It's a little bit hard to visualize, but I will try my best to show you. But I need more space. Hold on. Okay, so I am gonna zoom in here. I'm gonna take this thing, and then I'm gonna zoom in. Okay, zoom in. Zoomed in view of this location right here. Okay. All right. So this is the endoplasmic reticulum, right? It's like hollow on the inside. It's empty on the inside, okay? And then we're gonna have the ribosome here. Okay, that's a ribosome. Here, let me write this again. This is the zoomed in view. Just like a closer look, right? So over here, that is the ribosome. I know you guys have been like busy studying for the quiz and you might not remember everything from last class, but that's okay. I'll try to remind you, okay? The ribosomes are important for translation, right? Which is the process of making proteins. Right? And so the ribosome will build the protein. That's what this red chain is. Those little circles are basically amino acids. So that's the protein. Right? And once you make the protein and it goes into the rough ER, the protein is going to fold up and it's going to move towards the edge. And so that's like a folded up protein. And then it's going to move to the edge. And it's kind of like blowing bubbles, right? Like if you imagine you have the wand, right? You dip it in soap bubble and then you're blowing the bubble, okay? When you blow harder and harder and harder, eventually this thing is it's just gonna come off as a bubble, right? Can you visualize that process? Okay, so like blowing a bubble, it gets bigger and bigger and bigger, and then eventually it will just come off as a ball, right? And that's kind of like what's happening here. The protein will move towards the edge, and eventually it will pinch off, right, with the protein inside. And it forms what we call a vesicle. Vesicle, a vesicle pinches off from rough ER. And that vesicle is going to have proteins in it. Okay, so that's what's happening in the rough ER. Not only does it make the protein, using the ribosome, but it packages it nicely into this little bubble, vesicle. Okay? So you might have a bubble coming off here, and it's going to have some proteins. Right? You might have another bubble coming off here, another vesicle coming off, right? Okay. Any questions? Excuse me. All right. So let's take a look at what's going to happen to this bubble, right? So this bubble actually will move to another organelle. Right? So this organelle is also a series of membranes. Okay. And they are like arranged one after the other. It's like a stack of pancake. Right? And this thing is called the Golgi. Golgi body. And what the Golgi body does is it will sort, modify, and repackage proteins. Okay. So this vesicle that pinches off from the rough ER, it's going to go to the Golgi, right? And then it's going to fuse with the Golgi. Here, maybe I'll draw another one of these things right here. Oops, that's a little bit too big. It's going to fuse with the Golgi. And then the bubble is going to get passed forward and backwards a couple times. It's going to get sorted out. It's going to get modified. And in the end, you're going to repackage it into a new vesicle. Okay, and the protein inside is going to be a little bit modified. So let me try to find a different color, like maybe something like that. Oops, there we go. Right, so this is like a repackage vesicle. Right? It's like a similar same protein as before, but they made some modifications to it. Sometimes you can add sugar to the protein. Sometimes you want to cut it up a little bit. Right? There are a lot of things you can do to the proteins, which we're not going to go into. But all that stuff, all that modifications, all that sorting and changing, that's going to be happening in the Golgi body. Okay. Is everybody
Where's the
okay?
location of Golgi body?
Where is the location? It's somewhere in the cell. Okay. If this is the cell, this is the nucleus. The Golgi body might be somewhere here. Like it doesn't really matter, right? Like it's all over the place, right?
Thank you.
Yeah.
Okay, so for this RER does it also like, is it both in the plant and the animal cell, or what are we actually like? Is this a plant cell or the animal cell?
Yeah, yeah.
I'm kind of
Excellent question. This is just a eukaryotic cell, okay? Let's say this is animal for now. Okay, animal. Okay, but it's in the plant cell as well. Plants also have the rough ER. Okay?
oh,
Yeah, we'll talk about the
okay.
distinction between plant and animal cells later, but for now it's just like it's a eukaryotic cell. Okay. Now I'm gonna make the cell membrane longer here. Okay, just so because I want to show you a few more stuff. Okay, I guess we don't need this anymore, right? Like this stuff, it's just like a quick review. I'm gonna erase that. It's kind of in the way. Right, it's all in the notes anyways. Okay, but like everybody understand like where the green ball come from, the green circle, right? You have to, understand that. So in the rough ER, the purple lines, the purple tunnels, you have all these bubbles coming off. Okay, they're all coming off, and then they are not ready yet. That's why we need to send them to the Golgi, and then you're gonna modify it. Okay. Nika said it's very hard. Yeah, I know. But please ask questions, okay? Like I don't know what you don't know. There's no dumb questions, right? Just ask. If you have questions, I'm sure someone else also have the same questions. Okay, so help me understand how I can explain it better to you. Okay. So I'm gonna pause for a moment just to let you take it in. And if you want to ask anything, please do.
So Vincent, this ribosome is making like a protein. So the order of this protein comes from DNA inside nucleus.
That's right. So the DNA here in the nucleus, it contains the instruction to make the protein, right? Okay, so we talked about this last time as well, but let's just quickly write it down for you. The DNA has the instruction to make protein. It's the recipe, right? We're gonna make a copy of it in the form of mRNA. This is just copy of DNA, not the whole DNA, but just the section that gives you the instruction to make a particular protein. If you want to make insulin, then you copy the instruction to do that. If you want to make, another protein, actin, right? Then you copy the instruction to do that. Right? So that process is called transcription, right? Which happens in the nucleus. Once you have the RNA, then you're gonna use it to make the protein. And that process is called translation, which happens in the cytoplasm. Okay. So look, what is actually making the protein? What is actually building the protein? That is the job of ribosome, right? Ribosome helps make the protein. It reads the instruction from the mRNA and then it connects the amino acid together. Right? So we have ribosomes that are just floating on their own. They could make the protein, but those proteins are not packaged. Right? They are just proteins that you make, and they are available right away. Some of the ribosomes will be making proteins with the RER. Those ones will be packaged into a nice bubble, a nice vesicle, and the vesicle will get transported to the Golgi body. And then you're going to make some final changes, final adjustment to it. Okay? All right, moving on. I am sure you are curious as to what will happen to the vesicle. Well, the vesicle can do a couple of things. Okay? The protein that we make, sometimes they will move and become part of the membrane. Okay? Become part of the cell membrane. Right? So that protein that we make, the pink thing on the inside, they might become part of the membrane. Okay? We'll learn more about that later on. Okay? So it becomes what we call like a membrane protein. They are important for many reasons. For example, some membrane proteins give you your blood type. Okay. Or we can send the material out of the cell. Okay. So the bubble is gonna fuse with the membrane, and then the protein is gonna get released from the cell. Okay. Releases the protein out of the cell. And then you can use it for whatever purpose. Okay. Different proteins have different functions. You release it out of the cell, it's going to do its thing. Maybe you don't need it yet. So it will stay in the cytoplasm until it is needed. All right. And then there's one more possibility. It becomes another organelle called lysosome, which I'll explain in a bit. Okay. There are all these different paths that the vesicle can go once you have finished modifying it, finished sorting it, and so on and so forth.
Vincent, there is a confusion for me. So all these things happens in one cell.
That's
so
right.
when a function is going to be done, so a bunch of cells do the same process.
Yeah, so sure, right? So like, for example, let's talk about insulin again, right? You understand insulin? You remember we talked about that?
Yes.
Or yeah, so that's something that you need after a meal. If you eat food, you need to help absorb the sugar, and then you need to you need insulin to help you absorb the sugar. The insulin comes from your pancreas. Because the pancreas is like on the left side, right? Behind the stomach. So a bunch of cells in the pancreas is going to, basically all of them in the pancreas are going to undergo this process, and you're going to make a bunch of insulin, and then you will release it from the cell, right? But does all the cell do this? No, like your brain cell is not going to make insulin, right? Like your liver cell is not going to make insulin, right? It's only the cells that are specialized in making that protein. Those are the kind of cells that will do it. Does that answer your question, or that's not your question?
Yes, yes, I got it. Thank you.
All right, great. Okay, good questions. Anyone else? Are you guys with me? Can you give me some, feedback? Maybe like a thumbs up or something, or, it's really hard for online for me to gauge. I taught the same thing yesterday for like my in-person class, and then I can see some of them have like lost faces, right? So I was able to redirect the lesson, but here I have to rely on you guys. Okay, thank you. All right, so let's see. Let's go back to our PowerPoint for a little bit. So the next thing we're going to be talking about is this thing called the endomembrane system. We already drew a bunch of it, okay? Like I just didn't tell you that's what it was called because it was already way too much information at the time. But endo means within. Okay. Endo. Membrane is, we know what membrane is, like the cell membrane, right? Okay. So an endomembrane system is basically a bunch of membranes inside the cell. I don't know if you remember this, but I told you that the cell membrane is made up of phospholipids. And last time I told you there are other organelles inside the cell that also have a membrane, right? And we know this already. The cell, the nucleus has a membrane of its own, right? So there are a bunch of stuff that are made up of membranes, and they are inside the cell. So these network of membranes, they form tubes and sometimes sacs. Sacs is like a vesicle, like a circle, right? In the cytoplasm. And together they work with each other, and they will produce various things. They will produce lipids, they will produce proteins, and they will help, in some cases, to break down some of these larger molecules. And as you will see, it will also recycle and dispose of things that are no longer needed. So that's like an overview of the collective function of the entire endomembrane system.
Quick one. Sorry to take you.
No worries.
What is this cytoplasm? Where is it?
Okay, so this is a cell membrane, yes, and then the stuff inside is the cytoplasm. Okay, like the liquid. Think of a Ziploc bag. Okay, Ziploc bag.
Yeah.
You fill it up with some water. You sealed it. The water is the cytoplasm.
Okay.
Yeah.
Thank you.
No worries. Okay, so floating in the cytoplasm would be a bunch of these organelles, right? The ribosomes, okay? The nucleus, the Golgi body. Now, the endomembrane system, again, it's just a fancy way of saying a bunch of membranes inside the cell, include the following items. We know about the nuclear envelope. We know about the rough ER, the purple thing that I drew with a bunch of dots on it, right? The dots are the ribosomes. That's part of the endomembrane system. There's the smooth ER, which I haven't talked much about. We talk about the Golgi bodies. Sometimes you read online, they use another name, but it's the same thing, okay? Golgi bodies, Golgi apparatus, they mean the same thing. And then there's this thing called lysosome, which again, I haven't tell you much about it except for the fact that sometimes these vesicles, it can become the lysosome. Okay, I'll tell you what it does later. Okay, so basically everything we've drawn here is part of the endomembrane system. Right? The nuclear envelope, the rough ER, and then there's this pink stuff here, which we should label now. Okay, that's the smooth endoplasmic reticulum. Okay. It's smooth because it doesn't have the ribosomes, right? That's why it's like not bumpy, looks smooth. Endoplasmic reticulum, SER. I'll tell you what it does later, but I just want you to know that these are all the components of the endomembrane system. They are all made of membranes, right? Someone asked, I think it was Fatima, right? Asked like, what is it made up of, right? It's just membrane, phospholipid. Okay. All right, so let's go through a few more slides. These are things I already talked about in the picture, but let's go through them anyways. Rough endoplasmic reticulum, RER. On the next quiz, not today's quiz, but the next quiz, I will give you pictures and I will ask you to tell me what you are looking at. Please type the full name, okay? Rough endoplasmic reticulum. If you put RER, like the system will not recognize it. Okay, you have to type the full name. So what is it? It's a network of membrane that is continuous with the nuclear envelope, right? It's an extension of the nuclear envelope. And it's rough because it is covered with clusters of ribosome. Where do ribosomes come from? Does anybody remember? Where do ribosomes come from?
Nucleolus?
Very good. It is from the nucleolus, right? You make them there, they will exit through the pores. Either they are stuck to the ER or they're floating around on their own. Either way, their job is to make proteins. So that's a picture, right? These are like in the study guide, there are a bunch of pictures that you should know. Let me see where it is. Here, right? These are the pictures I will show you on the quiz, and you will have to know what it is. Okay, so like if I show you this picture and I point to this thing and you're like, oh, I see a bunch of dots there. This one is the rough endoplasmic reticulum. Okay? Of course, I'll post the answer, but it's you know, it's your job to make sure you you know, review them, right? Okay, so what does it do? The ribosomes would synthesize proteins directly into the rough ER, where it will be packaged into small vesicles that butts off or pinches off at the edge. This is what I was trying to draw for you, right? Like there's the ribosome on the outside, and then we see the peptide here. The peptide is going to fold up into the protein. It moves to the edge, and then you have the vesicle. Okay? Please tell me, where does this thing go next? Where is this going to go? What other structure?
The Golgi body?
Very good. The Golgi body, right? The pancakes, right? The orange thing that I drew. It's going to go there. It's going to get sorted. It's going to get modified and repackaged, right? But that's what the rough ER does. It's going to be responsible for making and packaging proteins along with the ribosomes. Okay. And then there is the smooth ER, which I only label for you. I haven't told you what it does. Now might be a good time. Why is this smooth? It's smooth because it has no ribosomes on it. Okay, it's just a distinction, like the name difference is based on how they look. If they have the dots, then it's rough. If they don't have the dots, then it's smooth. Without the dots, without the ribosomes, it's understandable that the smooth ER is not involved in protein productions. Right? No ribosomes, no proteins, right? Instead, the smooth ER is responsible for making lipids. We've learned a bunch of lipids, remember? In fact, that's going to be on your quiz, right? We learned about like triglyceride. We learned about phospholipids, steroids, right? So if a cell is involved in making steroid hormones, such as the ovaries and the testes, you would expect them to have a lot of smooth ER. Make sense? Let me say that one more time. We know steroid hormones are like testosterone and estrogen, right? So to make them, you would need the smooth ER. What kind of cells do you expect to have a lot of smooth ERs? Well, it would be the cells of the ovaries and the cells in the testes, right? Where they are making all these steroid hormones. The smooth ER also plays a role in breaking down fats. Not only do they make lipids, they will also break them down, and it will help detoxify drugs. Okay, detoxify drugs. In fact, if liver, liver is the organ that detox your blood, right? So when they are exposed to drugs, and I don't necessarily mean like, illegal drugs, it could be like medication drug, right? When your body is taking any kind of medication, your liver cells will actually increase the amount of smooth ER to help detoxify the compound, okay, to break it down, to metabolize it. Okay, so
Professor, excuse me.
yes,
Sorry.
go ahead.
I did not get well. What do you mean about detoxify of drugs?
Oh, so like
effect from drugs?
detoxify just means like okay, so sometimes
I
certain
know the
drugs
meaning. Okay.
drugs can be bad for the body, right? Okay,
Yes.
so then they need to break it down. They need to make it not toxic. That's what detoxify means.
Okay, it
Sometimes
means it's good. Break down
yes,
fat
it
and
breaks
drugs
down.
like
That's
that.
right. That's right. And even if you're taking good drugs like medication to help fight infections or something, you still need to break the drug down, right? So to break it
Okay.
down, it happens in the liver.
Okay, thank you.
Yeah. All right. So quickly again, nuclear envelope extends into the cytoplasm. Some have ribosome. That's the rough one. Some does not. That's the smooth one. What do the rough one do? Rough ER, they make proteins and they package it. Smooth ER, they make lipids, they break down lipids, and they break down drugs. Okay?
I have a question, Vincent. So this smoothie ER, they do only for example, the medication we took, for example, for anemia,
Sure.
or for all the medication they break down, or they have specific medication, for example.
So
for
generally, any kind of foreign substance that has like a medical property, it's gonna go to the liver and it's gonna get metabolized in most cases, right? So it doesn't really matter what kind of drug it is, but sometimes the kidney can help filter them as well, right? So like it's not like they do it for just one kind of drug. It's like a broad mechanism, right? Alcohol, for example, can be broken down in the liver as well, right? All right.
One question, Professor.
Sure, yeah.
Can you go back to the last? Okay. Is there any difference between ribosomes and rough ER, or the
Okay,
same, or they are one thing?
so the rough ER is rough because they have the ribosomes on it. Okay, so there are two different things. Okay, the ER, the endoplasmic reticulum, is just these membranes that are an extension of the nuclear envelope. Okay,
Okay.
they are like the nuclear envelope, but then they branches out. And if they
Okay.
have the ribosomes on it, then it's rough. If they don't, then it's smooth.
Okay, got it. Thank you.
All right. So then we talk about the Golgi body, right? It's several layers of membranes, and they receive the vesicles from the rough ER. not the smooth ER, okay? It's just processing the ones that have the protein. And as I told you, the contents are going to get sorted, repackaged, and then sometimes they get tagged, okay? They get like, they put like a, it's like a, you know, sorting center at a post office. They put a tag and they say, okay, this one goes to that city, this one goes to that city. Basically, the tags will tell the vesicle where to go. Does it go out of the cell? Does it become part of the membrane? Does it stay in the cell for later on? Is it going to become a lysosome, right? So basically, the tag tells it where to go, so they end up at the right place. All right. So here it says, where will the protein go from the Golgi body? I already wrote it down for you, right? Remember? With all the arrows.
Okay, so the Golgi body only works with the RER.
That's correct.
Okay.
All right. Very good. So this last thing, it will become a lysosome. What is the lysosome? What does it do, right? So lysosome is the last thing that is in the endomembrane system. Okay, remember that list, right? The nuclear envelope, smooth rough ER, Golgi body, and the last thing on the list is the lysosome, right? Okay, so here is a lysosome. My face is blocking it. There is a lysosome. It's like it's just a vesicle, but it contains a mixture of digestive enzymes. You might remember enzymes from lecture 2. They are proteins that help carry out chemical reactions, right? In this case, the enzymes are responsible for breaking down macromolecules. So if you need to break down a carbohydrate, the lysosome can do that for you, okay? So it breaks down macromolecules, and it also helps recycle old organelles. Sometimes some of the organelles gets worn out, they're no longer functioning properly, such as this one here. Okay, like you don't know what it is yet, you will, but this thing is not working anymore. So the lysosome will come, it will chew it up and break it down. Okay? The reason for doing that is we don't want to lock up resources, right? If we recycle the non-working organelles, we can get some nutrients back. Maybe we can use it to make a new one. When the cell picks up some food from the environment, right, from the surrounding, the food particle, maybe a macromolecule, is going to get, again, broken up by the lysosome, right? So now you get the monomers back. You can use it to build things that you need in the body. Okay, so think of the lysosome as like the garbage disposal recycling depot. Okay, things you don't want, things you want to break down, goes to the lysosome. Okay. Lysosome sometimes also plays a role, in programmed cell death. Okay, we call this apoptosis. Sometimes you want a cell to die in the body. For example, if the cell is getting too old, or if it's not functioning properly, you want to kill off the cell. You don't want it to stay there and, you know, cause trouble. So the lysosome will help trigger basically like the reactions that will cause the cell to die off. Okay? So this is deliberate cell death, programmed cell death. We call it apoptosis. Okay, so we'll come back to this later, but cancer cell has evolved in a way that they will not trigger apoptosis. They were supposed to die because they are not doing normal function, but they have changed so much in a way that they could evade apoptosis. Okay, so we'll learn more about that later. This is a picture of a fetus, 51 days old. and you can see that the fingers are starting to form. But did you know that there were webs between our fingers during embryonic development? And we don't have webs now when we were born. So the cells in the webs, they needed to die during embryonic development. So the lysosome will help trigger apoptosis in those cells. Get rid of them. So sometimes cell death needs to be deliberate, needs to be on purpose, for the reasons I've told you about. There is a group of diseases that are collectively known as lysosomal storage disease. Okay? So I told you the lysosome has a bunch of enzymes in it, right? And sometimes these enzymes might be missing because of mistakes in the DNA. Okay? Enzymes are protein. To make proteins, you need the DNA, right? So if the instruction in the DNA is wrong, then you don't make the protein. You don't make the enzyme. So depending which enzyme is being affected, the disease that the person have is going to be different. Most of these diseases are fatal in early childhood. Some, even though the person is gonna live on, but it's gonna have debilitating results for their life. So there are two examples here. Pompe disease is a situation where the lysosome is missing the enzyme to break down glycogen. You remember glycogen from lecture 2? That's the storage form of energy, right? So when you need to convert the glycogen back into glucose, you need to have an enzyme for that. If in this particular disease, you're missing that enzyme, so the glycogen will build up in muscle. Okay? And that's going to cause problem because it's going to weaken the breathing muscle, it's going to weaken the heart muscles, and it's going to cause cardiovascular problem. Okay. Another one is called Tay-Sachs disease. That's something that they sometimes will screen for in prenatal screening when you have a baby. And in this case, you are missing an enzyme that breaks down lipids. And those kind of excess lipids build up in the brain cells and interfere with brain development. Okay. So there are a bunch of diseases that are related to these enzymes in the lysosome. It's going to cause some problems.
Professor, lysosome is just mostly found in the babies?
No. So we all have lysosomes, right? Adults have lysosomes as well. But these things sounds like it's affecting the babies because when they're born and they don't have those enzymes, that's where the problem starts to occur, right? These are not diseases that you will get later on in life, like you are born with it, right? These conditions.
Okay.
Yeah. Good
Thank
question. All right, let's see if you remember this one. Let me launch the poll. Okay. Thank you. So if you look at the slides from before, ribosomes are made up of proteins as well as RNA. Okay, that's what they are made up of. What is the function of ribosome? To make proteins. Okay, but that's what they're made up of. here is another one. Okay. So to break down macromolecules, that would be lysosome. We literally just learned about that. And also the smooth ER, right? Smooth ER helps break down lipids as well as detoxification of drugs, right? That's all. So that is the endomembrane system. Before we move on, does anyone else have any other questions? Okay, remember to take the quiz today, huh? It will be available to you until 11:59 tonight. Only 6 of you have done the quiz. So, I mean, that's okay like that. I'm just reminding you.
Mr. Vincent, I have a question is about the quiz. I've done the quiz which was a self-assessment. It's not graded, but one of the questions says cell membrane is made of what, and I chose like phospholipid, and then I saw the right answer. It was protein. But you said to us that is lipid, right?
Yeah, it's lipid. Yeah, it's lipid for sure. The major component is the phospholipid. Which question is that? I'll take a look at that in the break, because sometimes the questions might, like the answer key might be wrong. Like we have thousands of questions in the bank made up by several people, and sometimes there might be mistakes. But I'll take a look during the break. Okay.
Thank you.
Yeah. All right. Other organelles. Let's keep going. Now there is this other thing. Okay, so everything we're gonna talk about now from this point on, these are not part of the endomembrane system anymore, right? Okay, we're done with the endomembrane system. Now we have a bunch of other things. And so there is this other thing called.
So what do we call these other things? What do we call them?
We don't have a name for them. They're just organelles. Organelles.
Oh, okay, organelles. Okay.
Yeah. So this thing is called peroxisome. Peroxisome. And you can see in this picture, it looks like almost identical, right, to the lysosome. Like how are you supposed to know? Like this circle is peroxisome, this circle is lysosome, right? Like one is darker than the other, but not always. So it's hard to tell, man. Like you have to really study these things to be able to tell them apart, which we're not going to worry about. Okay, I'm not going to ask you. I'm not going to give you a circle on the test and ask you if it's a peroxisome or lysosome. That there's no point of doing that. Okay, but they look very similar in the cell. Right? Like even in a picture, right? The only way you can tell is difference is like this one is green and this one is pink, right? So like I'm not going to ask you to identify them. But peroxisome is not part of the endomembrane system. They did not come from the vesicles, right? These are some created in some other ways. And the key thing is they carry out oxidation reactions. And oxidation reaction is a reaction where electrons are removed. Okay. So when you remove electrons from a molecule, you will help break them down. But the byproduct of doing that is something called H2O2. H2O2 is peroxide. Okay, peroxide. Like hydrogen peroxide, right? Hydrogen peroxide, actually, which you might recognize. It comes in a little brown bottle that you can buy from like Shoppers Drug Mart, right? It's sometimes used as a disinfectant, right? But H2O2 is damaging to cells. So we want to make sure we keep them inside the peroxisomes. We don't want it to come out. You can break it down into water and neutralize it, but you want the reaction to happen inside these peroxisome. And what it breaks down are things like fatty acids, amino acids, and even poison. And you might start to notice this sound very similar to what lysosome is doing or like smooth ER is doing, right? And it's okay. There are a lot of overlap functions going on in the body. Okay, so peroxisome is also responsible for breaking things down. The main difference here is that it is breaking it down by ripping electrons away from the molecule. Whereas in the lysosome, you are breaking things down with help of enzymes. Okay, so it's more like the difference is more like how you break things down. Okay, so that's the peroxisome. Here's another question. Okay, if you track the movement of a protein produced in the rough ER, it can end up in all the following locations except, so if you look at the notes that we took, right, like this one that we drew, like where does this thing go? Right? Well, it can go to become part of the membrane. It could be released out of cell. It can be in the cytoplasm, or it can become the lysosome, right? So what's the exception here? Well, it is not going to be inside a peroxisome. Remember, the peroxisome is not part of the endomembrane system. So that's why it has nothing to do with the stuff that are produced in the rough ER. Okay. Let's do one more before the break. The next one is called a mitochondria. Okay, this is plural. Singular is mitochondrion. Okay, just in case you see it and you wonder what it means, that's what it means, okay? So this is the site of energy production. Right? This is where energy is being produced. Remember, I showed you an equation earlier in lecture 2. You take the glucose and you react it with the oxygen to produce carbon dioxide, water, and energy. Well, part of that reaction is going to be happening at the mitochondria. Okay, so animal cells have mitochondria, plant cells have mitochondria, eukaryotic cells, they have mitochondria. Okay, so that reaction is called cellular respiration. Right, it was in earlier on in lecture 2. You can look that up if you want later on. Some cells have only a few mitochondria, especially if they don't have high energy requirement. Other would have thousands of them, okay, depending on the energy need, right? Can you give me an example of a cell type that needs a lot of energy?
Muscles?
Exactly, right? So like the muscle, you move your muscle around a lot. You walk around, run around, right? Your heart muscle beats 24/7 as long as you're alive, right? So all these places, all these cells, you would expect them to have more mitochondria. So there's a recurring theme here that I'm trying to present to you. All the cells would have all of these organelles. However, depending on the function, some cells might have more of one type than the others. I already gave you a few examples, right? Okay, write it down if you missed it earlier. For example, as we just said, the muscle cells might have more mitochondria because they need more energy. Okay. Do they have smooth ER? Yes, they do. But do they have a lot of it? Probably not, because its function is not to, make lipids, right? The function of a muscle cell is just to move, right? So it has more mitochondria. Right? Things like the cells in the ovaries, they might have more smooth ER to make the estrogen, to make the progesterone, right? So different cells have different percentage of organelles, right? Depending on what it does. So let's take a short break here, 5 minutes, and then we're going to draw more stuff when we come back. That would be 1:36. Okay, see you in a bit. So let's draw more pictures. All right. I don't know. Are the pictures helping you guys at all? Like some student like it when we draw things out. It helps them.
It's very helpful.
Yeah. Okay. Thanks. Yeah. If you don't like drawing, that's okay as well. You don't have to copy everything because I'm gonna post them anyway. Yeah, but I'm gonna do like a structure of mitochondria. Okay, remember ION is singular, IA is plural. Okay. So here we go. I mean, the reason we draw mitochondria and not like other stuff is we actually gonna come back to this in lecture 5. Right? We're gonna spend a lot of time talking about mitochondria in lecture 5. So that's why we need to have a good understanding of what it looks like. So it's gonna look like this. It's gonna have an oval shape. Okay, so this is the outer membrane. The outer membrane. When I say membrane, it's the same stuff that makes up the cell membrane, right? It's like the phospholipids. And then it's special because it has 2 membranes. There is another membrane on the inside, but the one that's on the inside, it's going to be folded up like this. It's going to be folded up like that. So this is the inner membrane. Okay, so like imagine a Ziploc bag inside the Ziploc bag. All right, so this is the inner membrane. Rather than straight, it's going to be folded up like this. Okay, and the reason is you can pack more into it. Right? So inner membrane folds up into these projections, which we call cristae. To increase surface area. Okay. Now, the space that's between the outer membrane and the inner membrane, the space between the outer and the inner membrane here, like this gap here, that is called the intermembrane space. And finally, the innermost portion, that's called a matrix. And all these parts plays a role in producing energy. And we're going to learn about that in lecture 5. But for now, that is what a mitochondria is. Okay. Questions? Okay. Done. All right, next we have this thing called the chloroplast. Okay, chloroplasts are found in plant cells, but not animal cells. Okay. It's possible to find chloroplasts in other cells as well. You guys know it's not just animal cells and plant cells, right? There are like other cells that exist, right? Like fungus, for example, like those freshwater living things that I told you about, the amoeba and the paramecium. So those are not animals, those are not plants, they belong to some other categories. So it's possible for some of these other cells to have chloroplasts, but when it comes to animals and plant cells, only plant cells have it and not the animals. This is the site of photosynthesis. The site of photosynthesis. If you don't know what that is, I will show you on the next slide. It contains chlorophyll, which is the stuff that makes plant green. If the plant part is green, then it contains chlorophyll, and that's the compound that allows the plant to capture the sunlight and use it to make food. Okay. Much like the mitochondria, it also has two membranes. You can see there's an outer membrane and an inner membrane. Never mind these names, okay? I'm not going to ask you for them. So that's the chloroplast. Some people buy chlorophyll extract and add it to their smoothie or something. There's no apparent health benefit for doing that. It's just a gimmick. If you eat your food, like if you eat the green vegetables, you are getting chlorophyll anyways. Okay, things that are a little bit darker might have more chlorophyll, but then there's no health benefit in, you know, in absorbing more chlorophyll. Okay? You eat the green vegetables for the fibers, right? Not for the chlorophylls. Okay, this is an important slide. Important. Okay, on quiz number 2, one of the written response questions will be based on this slide. Okay, like today's quiz, there are 2 questions where you have to type answers, right? Okay, on the next quiz, you will also have that. And one of the questions will be based on this slide. Okay, so listen to what I have to say. This is a chloroplast. Chloroplast is where photosynthesis takes place, right? Okay, photosynthesis. What exactly is photosynthesis? Well, photosynthesis takes carbon dioxide and water, and using sunlight, it is going to convert it into food, into glucose and oxygen. So this glucose is going to be stored as starch, right? Storage form of energy. So you see, glucose and oxygen. And then the glucose can be burned, can be used as fuel by the mitochondria. And the reaction that's going on in the mitochondria is called the cellular respiration. And in this case, you are taking the glucose and you're reacting with oxygen to produce carbon dioxide and water. You see, the ingredients for photosynthesis are the products of cellular respiration. And the ingredients for cellular respiration are the products from photosynthesis. So these two processes complement each other. Okay. So plants have the chloroplast. They make their own food, and then they burn their own food to make energy. Okay. Humans, we do not have chloroplast, so we eat the food. We eat the starch, right? We consume carbohydrates, and then we burn them for fuel. And that's why plants are happy without humans. They are self-sustaining. They could make their own food, and then they could, burn the fuel in their own mitochondria. Whereas humans, we are reliant on the plants. Okay, we cannot make our own food. We must consume them. Okay, so this shows the relationship between chloroplast and mitochondria. Does anybody have any question about this slide? Okay, so make sure you have a good understanding of it. Make sure you can describe the relationship in a nice way. Okay, you don't have to. One of the mistakes people make on the tests and quizzes is they try to use really big terms to explain things, and it just end up sounding a little bit strange. You don't have to use complicated terms. Okay, just use simple English, and you use the appropriate terminology when needed. Okay, for example, if you need to say photosynthesis, that's okay. That's a appropriate use of the terminology. But don't try to search things up online and then start copying with like random stuff. It sounds really out of place, and it's just not going to get you good grades. Okay. All right, here is another plant exclusive organelle. Okay. So chloroplast is plant exclusive. Central vacuole also plant exclusive. So this is a plant cell, this whole thing, and this whole thing in the middle. Okay, this whole thing here, that's what's called a central vacuole. Okay, a vacuole is like a big sac, and in this case is filled up with mostly water and some nutrients. Okay. In some parts of the plant, the central vacuole might contain pigments or colors for the flowers, and even contains poison for some plants. Okay. So it's just like a place to store stuff, essentially. Water and nutrients, primarily. Okay. Sometimes color, sometimes poison. Obviously, not all plants are poisonous, so they're not going to be all storing poison. But some are, right? Like a poison ivy. There is an other kind of vacuole, right? This is not in the plant, okay, but it's gonna be found in some single cell organisms, like this guy. You might remember this picture. This is a paramecium. Okay, it's the same category as amoeba. They both live in fresh water, fresh pond water, as opposed to like seawater, right? That's what fresh means. And one of the challenges of living in fresh water is that the water are constantly coming into the cell. And if you don't pump them out, it's going to cause the cell to swell up, and then it's going to pop. Okay. So that's why they have a contractile vacuole, which is this thing right here. It constantly squeezes and pumps the water out. And that prevents the cell from bursting. Okay, prevents it from popping, essentially. Plants also have cell wall, right? If you study for today's quiz, you will know cell walls are made up of cellulose, right? So cell wall is found, oh, it says right here, okay, but that's okay, you can write extra time. The cell wall is found outside of the cell membrane. And unlike the cell membrane, it is not selectively permeable, meaning it doesn't control what goes in and what goes out. Everything can pass through the cell wall, you know, without much regulation. The point of the cell wall is just to protect the plant cells from bursting. Okay, that is all. Okay, let's see if you can do this question. Okay, it's been more than a minute. I think I'm gonna stop that. So let's take a look. We're looking for the false statement. Okay, things that are not true, huh? Smooth ER is part of the endomembrane system. That is correct. Okay, remember endomembrane system includes the rough, smooth ER, the lysosome, the Golgi, and the nuclear envelope. There is an abundance of mitochondria in muscle cell. That is true. Abundance means a lot of, right? Muscle needs to contract all the time. It needs a lot of energy. All cells have cell wall to protect itself from the environment. That is not true. Do we have cell walls? Absolutely not. Only plants do, right? Yeah, we do not. Hydrogen peroxides are found in lysosome. That is not true. It is found in the peroxisome. Peroxisome. Oops. Chlorophylls are found in chloroplasts. That is true. Okay, so the only two statements that are not correct are C and D. All right, we are almost at the end of the lecture. Next thing we're going to learn is cytoskeleton. So cytoskeleton is a network of fibers that are found throughout the cytoplasm. You don't have to draw this, but I just want to show you what I mean. Like, how does the nucleus stay in one place? How come it doesn't, like, you know, get bounced around every time the cell moves, right? How do these things stay in place, right? How does the vesicle go to the Golgi body? Does it just randomly move and hopefully it will hit the Golgi body? Well, it turns out, and again, you don't have to draw this in your picture, it's going to ruin it, but it turns out inside the cell, you have all these fibers. Okay, all these fibers that are in the cytoplasm, and they are going to hold things in place. Think of it as like a giant web, like a spider web maybe, and then you can stick things here and there. Okay? And there are actually tracks that these vesicles can travel along, right? And the tracks will take them to different places. And so all these network of fibers that I drew here, those are called the cytoskeleton. Okay, so they are primarily there for support and movement. We have three types here: the microfilament, microtubule, and intermediate filaments. Okay, so I'm just going to show you what each one does and give you some concrete examples. Let's start with the first one. Microfilament. So this is a cell. This whole thing is a cell, okay? And I told you several times, cells are colorless. Okay, they are transparent. Okay, but you can highlight them with different colors under the microscope, okay, to highlight certain features. So the red thing here in the center, what do you think that is? You must know, right? What is that?
Nucleolus.
Close. That's the nucleus. Yeah. We can't really see the nucleolus. Probably is inside.
Yeah.
Okay? Yeah. That's the nucleus, and all these green stuff, all these green lines, those are the microfilaments, right? And so you can see it's all over the cytoplasm. So what do they do? First of all, what are they made up of? They're made up of this protein called actin. Okay? It's just a name. And they provide attachment sites for various organelles, which is what I've been telling you, right? Let's say you have a mitochondria, right? Like you can stick them on. Right? Without the cytoskeleton, then they just flow around randomly, bump into each other, and that would be chaos. But as you learn, organization is a feature of living things, right? So it's not surprising that you have these microfilaments to help organize things. The microfilaments can also help with movement. Let's say this cell right here on the screen, it wants to move that way. It wants to move to the right, for example. Then what can it do is it will break down all these microfilament, and then it will push it to one side. Right? So when it pushes the microfilament to one side, the whole cell will shift in one direction. Okay. So by reorganizing the microfilament, the cell is able to move around in the body. Okay. So microfilament provide attachment sites and allows the cell to move. So that's what you should be doing, right? When you are studying, you want to fill out the study guide. After you do the quiz today, you should probably do this tomorrow. Okay, write out key things that you should remember for each organelles. Okay? And again, it's okay to have your study guide with you when you are doing the test or the quiz. It's not a problem. Okay, so the second cytoskeleton is called intermediate filament. And so this is another picture. It shows you the intermediate filament, and you're like, that just looks exactly like this. Okay, and you're not wrong. Right? These fibers are overlapping. Right? Having one does not stop you from having the other one, right? Okay, they are just like layers on top of each other. And in this case, the intermediate filament, not only does it provide support within the cell, but it's also able to form intercellular junction. What does that mean? Let's say we have a cell here. Where is my mouse? Okay, it's right here. So there is a cell, and then there is another cell. Okay, and sometimes in parts of the body, you want to lock these cells together. You want to stable them, glue them together. And that's when you're going to need the intermediate filaments. For example, in your intestinal tract, right? In your intestines, you want to have these tight seals. Otherwise, the food is going to leak out of your cells and go all over the place, right? We don't want that, right? So sometimes you need to form these intercellular junctions, like connection between cells. That's what it means. The intermediate filament also plays a role in stabilizing the nuclear envelope. Here is the nucleus. And you can see the fibers are more like denser, higher density compared to out here. Out here, it's like kind of loosely connected, but when it's around the nucleus, it's really, really dense, right? So that will help stabilize the nuclear envelope. Of course, that's important, right? Because the DNA is inside the nucleus, right? If the nuclear envelope becomes unstable, then the DNA can be exposed and you could damage. There's a genetic disorder that causes a type of intermediate filament called lamin A to become unstable. Okay, and because of that, the nuclear envelope is unstable, and it causes this condition called progeria. Have you guys seen pictures like this before, where a person looks old, but then at the same time, it's kind of looks like a, there's certain child feature to them as well? Has anybody heard of these before? Just wondering.
Yes.
Yeah. So this is accelerated aging. Okay, it's called progeria, and it's because of the unstable intermediate filament around the nucleus. Okay. The person here is only about 9 or 10, but he has all the features of an aging person. There is currently no cure for this kind of disease. Okay, it's very, very rare. If you want to learn more about it, you can search it up yourself. Okay. Finally, we have the microtubules. That's the third type. The first one was microfilament. Second one, intermediate filament, and the last one are microtubules. So back to my question here, right? Like, how does the vesicle over here, how is it able to find the Golgi body? This is going to be hard to believe, but trust me, this is the truth. It does happen. You have a molecule that looks like a little person. It looks like a person without a head, okay? But it has arms, and then it has legs. And then it will grab onto the vesicle like the picture shows here, and then it will walk on these microtubules. Okay? It's like carrying the vesicle, and then it's just going to be walking along the microtubules, and then it will carry the vesicles to the Golgi body. So the microtubule provide tracks for these vesicles to move along. If we rely on random movement, it's never going to get to where it needs to go in time. Okay, so these kind of movement has to be deliberate. Okay, and that's what the microtubules are there for. The microtubules can also provide a little bit of structural support. Right, so basically all of them helps with structure and stability. Okay, but in this case, it also helps move vesicles along. This is true. You can actually search this up on the internet.
So Miss, can you, okay, this kinesin,
Yeah.
part of microtubules?
They are part of, oh no, they are proteins. Okay, these are proteins, but the protein walks on the microtubule. Yeah.
Okay.
Mm-hmm. Okay.
What about the ATP?
Yeah, we haven't talked about that, but for now, just think of that as energy.
Okay.
Yep. So, nothing's free. To move along the microtubule, you need to supply energy.
Okay.
Yeah.
Professor, excuse me. Why you say the protein is kinesin?
Kinesin? Yeah. No, someone was just asking what they were asking if this is part of the microtubule, and I said no. This is just a separate protein. Yeah. You don't have to know this name. Okay. This is not important for the test purposes. Okay. Yeah, there you go. But there is this thing that walks around, okay, with the vesicle. That is truth. Okay, just a few, I think 2 more slides, and then we are good to go. Centrioles. This is important for animal cells. Right? It's not found in plant cells. The centrioles, they make microtubules. They make a special kind of microtubules called spindle fibers. Okay, now there are a lot of terms here, but again, never mind them. You don't have to memorize it. Okay, the key thing to know here is that the centrioles, they are going to send out these spindle fibers, which will help move the chromosomes around during cell division. Right? We're gonna do more of this later on in lecture 7. When a cell divides, you need to tell the chromosomes to go here and there. Okay, but again, we don't want like random movements. They might get tangled up. They might not end up where they're supposed to. So these microtubules that are produced by the centrioles, they will help guide their movements. Okay, it will take them where they need to go. Some structures, there are other structures that are made up of microtubules, like the flagellum. Okay. Flagellum is singular, flagella is plural. So usually there's just 1 or 2 per cell. And in humans, the only cell that has a flagellum are the sperms. This is the sperm. Okay, that's the only place where you will find the flagellum. It helps them swim, right? And only 1 tail for the sperm. Other cells, like not animal cells, like some other cells, they might have 2 tails sometimes. But only 1 or 2. Cilia usually covers the entire cell body. You know that paramecium I keep on showing you with a vacuole, right? That has cilia all over the cell. Okay, it has the vacuole here. It also has a mouth here for real. So all these hair just beats and help it swim around. In humans, we also have cilia. Right? This is the airway. Okay. When you breathe in pollutants and dust particles, the cilia in the airway helps sweep them up, and you can cough them out. Okay. This is somewhere in the female body. Anybody want to guess where that is? It has some cilia in it as well. Only in females.
The fallopian tube?
Excellent. This is the fallopian tube. Okay, also known as the oviduct. So the egg is released by the ovary, and these cilia is going to tell it where to go, right? Like, go that way is the uterus. Okay. So cilia and flagellum, they are both made up of microtubules. This is a summary that shows you what the difference are between the plant cells and the animal cells. So for example, plant cells, they have central vacuole for storing water, nutrients, sometimes poison, sometimes color. They have a cell wall. Humans, we have, what do we have? We have the lysosome. The plants don't have them. Okay, it says centriosome here, but we call it centriole. There is a slight difference, but we don't care. It's already too much information. Okay, so there are some things that are found in animals and not plant, and some are found in plants and not animal. Okay, you can go through the list later. Last question. Which of the following is made up of 2 intertwined actin protein strands? Okay. So I'm just going to show you. I think you guys are, running out of steam. Right here. Right. Microfilament made up of 2 intertwined actin protein strands. So the answer is microfilament. Okay. And that's it for lecture 3. All right. I know you have the quiz to worry about, but if you have time before our next class, please do this thing. And at the beginning of next class, I think I'm gonna have some time, and I'm gonna label this for you. You can try to label it yourself, okay? But we'll label it together at the beginning of next class. For example, what is this thing? Does anybody know after today's lecture? What is this?
Mitochondria.
Yeah, exactly, right? Mitochondria. Right? And it makes energy, right? So practice doing this, label as many things as you can. I mean, there are some words here, but you know, label as many things as you can, and write down their functions. Okay? Well, thank you for,
Professor,
yeah, go ahead.
oh, sorry.
No worries, go ahead.
This is just one cell?
Yes, this is one cell.
One cell.
It's an animal cell.
And all the things in cells is just these are.
That's right. All the
Okay.
organelles, right? Here is a plant cell, right? Like, for
Okay.
example, what is this thing here? Anybody?
Vacuole.
Central vacuole.
That's right. The central vacuole, right? Where you store the water and the poison and the color, right? Okay. If you say it enough times, it will stick in your head. Okay. If you write it out enough times, it will stick. I know it's a lot of information. I really do. Okay. I'm not saying it's easy. But hopefully, you know, with the study material and the recordings, right? You can watch the lecture again if you need to. All right. So good luck with your quiz. I will post the result, you know, tomorrow. One thing about the quiz, though, I actually want to tell you.